Hello everyone. Today we will be discussing on Neisseria gonorrhea, which is also commonly called as gonococcus. Going to the next slide, where we are going to study about the characteristics of Neisseria gonorrhea. There are certain characteristics of Neisseria gonorrhea which we should know before going into the morphology and other parts of this, before discussing the morphology and other characteristics. So what are the important things that we should know before beginning the morphology? That is that this can grow and rapidly multiply in mucous membranes of humans, and where it can grow and multiply in the mucous membranes of mouth, throat, anus of males and females, cervix, fallopian tubes, and uterus of the female reproductive tract. So these are the places where it can grow and multiply rapidly. And these are the mucous membrane of the place where it multiplies, mucous membrane of all these which I have mentioned. And this bacteria can be transmitted through sexual contact. So it is a venereal disease. It causes venereal disease, and it can pass through sexual contact, that is through vagina, oral or anal contact, and can spread from mother to child at the birth. How all this happens and what is the pathology, that we are going to read in the next video. In this video, we are going to discuss about the morphology. So coming to morphology. Shape oval or spherical with adjacent sides concave. I'm going to show you that shape also. So the adjacent sides are concave, and this I will be showing through a figure. While it's arranged singly or in pairs, usually it's a diplococci which is present in pairs, and its adjacent sides, concave sides, are adjacent towards each other. I'm going to show you this in the diagram. It will be more clear. And it is usually present inside the cell, intracellularly present. It requires oxygen, hence it is aerobic, but it can also survive in anaerobic medium. This I'm going to show you when we will be studying its culture. Then when we are doing gram staining of this, it is gram negative cocci, and as the gram negative is usually the ones which look pink or reddish in color because they take eosin dye. So I'm going to show this in with the diagram too. It's non-spore forming bacteria, non-flagellated, and it's hence non-motile. It's capsulated, and this diplococci is enclosed inside the capsule, which can be seen inside cells. And pili is also present, and usually this pili is present in virulent forms only because it helps in adhesion to the mucous membrane. So this pili is a strongly used for adhering to the mucous membrane of the places which I have mentioned in my slide before this slide, right? In my previous slide. So coming to the microscopic view of this Neisseria gonorrhea, it's when we see through electron microscope, we can see that the adjacent sides are concave, which are attached to each other. This is a diplococci, so it is arranged in pairs, and it has a pili all over it, which helps in adhesion. And that is why once it adheres to the mucous membrane, it multiplies very rapidly. And its outer membrane, this you can see it's a gram negative bacteria. How we can be sure about it? By performing gram staining. And here in gram staining, these diplococci are seen inside the cell. So this is gram negative, which are seen inside the cell, intercellularly present, and it has pili-like structures also. So coffee beans shaped, you can call it coffee bean shaped, as it's concave, and its adjacent sides are concave, which are attached to each other. Coming to culture media and culture characteristics, it can grow on chocolate agar. And here you can see this is Neisseria gonorrhea growing on chocolate agar. It can grow on Muller-Hinton agar, and this is the colony of Neisseria gonorrhea on Muller-Hinton agar. What are the different characteristics? How you can differentiate this colony? That we are going to read in the next slides. This can also, Neisseria gonorrhea also grows on modified Thaya Martin media, which is also called Ramal agar media. This is basically chocolate agar media, which is modified by adding four different antibiotics. One is vancomycin. This vancomycin inhibits the growth of gram-positive bacteria. Another one is colistin, and this this colistin inhibits the growth of other gram-negative bacteria. That means we are making a selective media for Neisseria gonorrhea. So we are adding colistin, so it inhibits the growth of other gram-negative bacteria, while Neisseria gonorrhea is a gram-negative bacteria, but it can survive because this is a selective media, selective and inhibitory media. It is inhibiting the growth of gram-negative bacteria, gram-positive bacteria. Then we add nystatin, which inhibits the growth of fungus, that is yeast and molds. And then we also add trimethoprim. This is to inhibit the protease species, swarming protease species. So protease species will also not be able to grow on this. So what are the colony characteristics of this bacteria? Its colonies are thin, round, translucent, convex, or slightly umbonate with finely granular surface. Lobate margins are there after incubation of 24 hours. You can see the lobate margins in this. What are the different culture characteristics where you can differentiate it that, yes, this is Neisseria gonorrhea? See, we have added trimethoprim in this to avoid protease species, because protease species also causes urinary tract infections. So that is why, to isolate this bacteria, this I'm talking about when I was talking about Thaya Martin media. I forgot to discuss this. So there we have added trimethoprim to avoid protease, because protease is also one of the leading cause of UTI infections. So while we are isolating the specimen, while we are collecting the specimen, it may get contaminated with protease also. So when we are culturing, we want to culture Neisseria gonorrhea, we have to add trimethoprim. Now, coming back to culture characteristics, this readily grow in media containing blood or serum. That is why we have taken chocolate agar in this case. Optimum temperature required is 35 to 36 degrees centigrade. Optimum pH is 5.8 to 8, but it grows very well in the neutral kind of pH, that is 7 to 7.2. It is aerobic, but it can grow anaerobically, that I have discussed with you. And it's good to provide 5 to 10% carbon dioxide to this culture media. And it's difficult to grow in laboratory as compared to Neisseria meningitidis. We will be talking about Neisseria meningitidis in the coming videos, but remember that it is more difficult to grow in laboratory as compared to Neisseria meningitidis. It has a complex nutritional requirements, and it can be differentiated into 4 different type of colonies, that is type 1 to type 4. Type 1 and type 2 forms small brown colonies. So brown color colonies are there, and they are pillated and are wide. Since I have told you that pili is used for adhesion. So if the that is Neisseria gonorrhea, if adheres to the mucosal walls, it is even while micturition, it is not thrown out. So this pili adheres very strongly to the mucosal membranes, and that becomes a major cause, because once adhesion is not, it is strongly adhered, then it grows and multiply there, and so it becomes virulent also. So it's more virulent when pili are there. And type 3 and type 4 are non-pillated, so they form smooth suspensions, and they are avirulent also. So this was all about the morphology and culture characteristics of Neisseria. In later videos, we will be talking about pathogenesis of Neisseria gonorrhea. Thank you for patient hearing.